Okay, welcome. Thank you for joining us. I'm here to interview Jason Carmel about his exciting work that he's presenting at a poster session here at the ANA meeting. So Jason, why don't you tell us about yourself and your training and your interests?
So I'm a child neurologist. I did my neurology training at Columbia, and I'm also a PhD interested in the motor systems. So my current position is assistant professor at Cornell, and I run my lab at a rehabilitation institute called Burke in White Plains.
So, and your abstract was in the rehabilitation category, and it deals with plasticity and recovery of function after brain injury. So why don't you talk a little bit about what kind of question you were trying to address with your work?
So we see brain and spinal injuries as essentially a disconnection syndrome. The brain centers that control movement are disconnected from the spinal centers that execute those movements. So after brain and spinal injury, there's almost always some sparse connections between the brain and the spinal cord, and we thought to strengthen these connections. This is an approach that others have taken. The innovation, or the different approach that we have, is to use the language of the corticospinal system, or other neural systems as well, which is electrical stimulation, in order to provoke outgrowth at the level of the spinal cord and to strengthen connections using activity-dependent plasticity. A different sort of activity-dependent plasticity than I think most neurologists are familiar with. We think of activity-dependent plasticity as changing synaptic functions, making them stronger. The particular anatomical locus for our changes, we think, is at the terminations, where the corticospinal tract actually terminates within the spinal cord gray matter. Those connections sprout out and form stronger connections, and that has behavioral relevance for the animals.
Sprouting of the axon terminals. So your work is very interesting in this instance because you took a chronic injury model and tried to see if a chronic deficit could be reversed. So why don't you tell us about the model that you used?
So there was a sort of empirical and a scientific question that we were trying to figure out. The empirical question is, obviously most of the patients who we see are chronically injured. They've had neonatal brain injury, they've been involved in accidents. For adult patients, they've had an ischemic stroke. And so months to years afterwards, can we actually recover function in these people? Scientifically, we've looked previously at the separate effects of activity-dependent plasticity and injury-induced plasticity. Injury itself causes sprouting at the level of the spinal cord gray matter. That gets augmented with stimulation. Does stimulation have to be presented at the time of injury? In other words, do those processes have to happen simultaneously, or can it be added on later? And so that was the core scientific question. So in rats with partial corticospinal injury, we stimulated the intact fibers. So we cut all the fibers from one side and then electrically stimulated on the intact side, with hopes of promoting connections between the same side of the brain and the spinal cord, right hemisphere to right side of the spinal cord. In other words, can we improve the right hand in humans, or the right forelimb in rats?
And you started about 2 months after the injury.
Right. So this is in humans you might call this subacute, but in the animal world this is certainly falls in the chronic category. And so these rodents have a stable deficit of forelimb control as we measure their walking over an irregular surface where they have to take sensory information in and move it into a motor program. When we apply the electrical stimulation, which we do using electrodes implanted over motor cortex, we stimulate in order to provoke a movement in the contralateral forelimb. And what we see is we stimulate each day 6 hours a day for 10 days. And about 2 weeks later, we start to see a decline in the number of errors as they're walking over this irregular surface. That functional deficits continues to get better even beyond the stimulation period. So while we see anatomical outgrowth at an early time, say 10 days, the functional effects continue to improve. The function continues continues to improve, and the function persists as long as we've looked out. So from the time of stimulation, at least 2 months later.
Wow. So this seems like it would have really exciting translational implications. Can you kind of talk about
Right.
what you think those are?
Absolutely. So humans with these types of injuries, brain, spinal, strokes, have a similar disconnection syndrome. We think that we can make hand impairments better by strengthening the intact circuits after injury. How we do that in our rodents, we put electrodes on top of the dura. We see a dual strategy. One is using non-invasive brain stimulation, not for alteration of cortical excitability, which has been its main use in the stroke field, but actually as a trophic effect on spinal axon terminations. The other would be to actually use a very similar strategy to our rodents, targeting specifically the impaired representation and delivering this electrical stimulation to promote connectivity.
So with an implanted stimulation.
With an implanted device.
That's really fascinating. So, and at your institution, you're doing animal studies, but there's a lot of human rehab research. So
right.
Do you plan on moving forward in the near future, or what do you see happening?
So we've the first trial that we were planning is to use a non-invasive form of stimulation called transcranial direct current stimulation. This is in children with congenital hemiplegia, and we're treating them as they're getting into school age. So most of their injuries happened before or at the time of birth, and so that would be our first trial. But we're certainly interested in looking at other strategies of delivering electrical stimulation safely and effectively.
Well, that's really exciting. I want to thank you for your time and sharing your work with us, and I want to thank everyone for your attention. So.
And thank you to the ANA. In addition to this opportunity, they've also given opportunities for young investigators like me to learn from department chair about how to become an independent investigator. It's been an invaluable experience.
Great. It's great.